Hello knowledge seekers, let me welcome you all for another video session on the topic of metabolism of carbohydrates. So in our discussion today, we'll be learning what are the various steps or various reactions involved in glycolysis. So majorly, as I discussed yesterday, the entire glycolysis constitutes of 10 major reactions which are divided or which can be divided into three phases. The first phase is energy investment phase or priming stage. The second phase is splitting phase, and the third phase is energy generation phase. As the name itself indicates, the first phase is where the energy is consumed for the reactions to occur, and in the second phase we observe a 6 carbon compound being split into two molecules of 3 carbon compounds. And in the third phase, as the name indicates, it is the energy generation phase. That means the glycolysis process eventually results in production of ATP molecules, which is nothing but energy currency of cells. So let us dive deep into each reaction and observe what actually happens in those reactions. So to start with the very first reaction in the first phase, that is a phosphorylation reaction. So the energy investment phase begins with the first reaction, phosphorylation, where the glucose which has been uptaken or which has been brought into cells through two different pathways, which is one insulin dependent pathway where the GLUT transporters act in bringing glucose molecule which is bound to insulin, and the second pathway is insulin independent pathway where the natural carriers acts in bringing the glucose molecule from the outer environment into the inner cellular cytoplasm. So in this reaction, glucose molecule is converted to glucose 6-phosphate in the presence of enzyme hexokinase or glucokinase. So what is the importance of this reaction? So this reaction occurs in, so hexokinase or glucokinase requires magnesium ion as a cofactor for their activation here. So this hexokinase enzyme is observed in almost all cells of the body. So it has low km value. That means a km is Michaelis Menten constant. So low km indicates high affinity for a particular substrate. So hexokinase has a low km value for glucose. That means when glucose concentration is just at zero point 0.9 millimoles, the enzyme gets activated to act over glucose present at that low concentration itself. So it has low km for glucose, so it converts glucose to glucose 6-phosphate. So hexokinase and reaction of glucose conversion to glucose 6-phosphate is a best example of feedback mechanism. Also, why I'm telling you this is, see, the glucose 6-phosphate produced at the end of this reaction acts as a natural inhibitor for the hexokinase activity. Now let's see about glucokinase. So glucokinase basically is present in liver cells. So in liver cell, glucokinase acts in conversion of glucose to glucose 6-phosphate, where it exclusively acts over glucose in its conversion of glucose 6-phosphate, whereas hexokinase acts over other hexose molecules like mannose and lactose. I'm not sure about lactose. Let me get back to you with that. I'm sure as far as mannose is concerned. So as I'm discussing, glucokinase has a high Km value. That means low affinity for glucose. That means a minimum concentration of 10 millimoles of glucose is required for the activation of glucokinase enzyme in the even the liver cells itself. So if you look at what other importance can we find in this reaction is, yeah, so the formed product glucose 6-phosphate is a primes molecule for other pathways also. I mean, for example, so it can act in glycogenesis, it can act in so we observe the presence of glucose 6-phosphate in gluconeogenesis. We observe its presence in other pathways like pentose shunt pathway also. So that is the importance of this glucose 6-phosphate, which is formed in the first reaction, which is first reaction called phosphorylation reaction of glycolysis. Okay, the second reaction is the glucose 6-phosphate, which is formed in the first reaction, undergoes an isomerization reaction in the presence of enzyme phosphorus hexose isomerase to form fructose 6 phosphate. That means glucose 6 phosphate is isomerized, undergoes is isomerized to form fructose 6 phosphate in the presence of enzyme phosphostriose isomerase. So it is a kind of a reversible reaction. And the first point is like, and the major point we have to remember is the first reaction where glucose is converted to glucose 6 phosphate, one molecule of ATP is consumed. Hence it comes under the energy investment phase of glycolysis. So one molecule of ATP is consumed in conversion of glucose to glucose 6 phosphate. Uh, Later, the glucose 6 phosphate is converted to fructose 6 phosphate in the presence of enzyme phosphostriose phosphorus hexose isomerase enzyme. So the next reaction is fructose 6 phosphate is converted to fructose 1,6 bisphosphate by consumption of another molecule of ATP. So another molecule of ATP loses one phosphate, uh, or donates one of its phosphate, forming ADP or turning into ADP, resulting in the formation of fructose 1,6 bisphosphate from this reaction. So this reaction is an irreversible reaction and is catalyzed by the enzyme phosphofructokinase. So, and this is a regulatory step in glycolysis. This is a regulatory step in glycolysis, and why this is a regulatory step in glycolysis we'll be learning about it in later videos. I'll be explaining it in the next part of the videos in detail. So the three reactions of the energy investment phase or priming stage has been completed now. So now let us look into the splitting. Now we are entering into the splitting phase. So in the splitting phase, what happens is still now, if you observe, starting from glucose to the fructose 1,6 bisphosphate molecule, all the molecules formed are 6 carbon compound molecules. So now for the first time, now one molecule of 6 carbon compound will split itself and forms into two molecules of a 3 carbon compound. So the first reaction of splitting phase is a cleavage reaction. So in this reaction, what happens is fructose 1,6 bisphosphate cleaves itself or gets broken down into two molecules. One is dihydroxyacetone, which is abbreviated as DHAP, and the other molecule and another molecule of glyceraldehyde 3 phosphate, G3P. So this reaction is catalyzed by uh, aldolase, or simply aldolase, to be precise, phosphos fructose 1,6 bisphosphate aldolase. So this enzyme catalyzes this reaction of this cleavage reaction, where DHAP is from one molecule of DHAP is from, and one molecule of glyceraldehyde 3 phosphate is formed. So the next reaction in the splitting phase is an isomerization reaction. What happens in this isomerization reaction is, so DHAP or dihydroxyacetone phosphate is reversibly converted to glyceraldehyde 3 phosphate in the presence of enzyme um, phosphostriose isomerase, in the presence of enzyme phosphostriose isomerase. If you remember properly, so glucose 6 phosphate is converted to fructose 6 phosphate. It is also an example of isomerization reaction. There the enzyme acted as phosphorus hexose isomerase, because the compounds there is a 6 carbon compound. Here the compound, so DHAP is reversibly converted to G3P, which is glyceraldehyde 3 phosphate, and the enzyme which catalyzes this reaction is phosphostriose isomerase, because here the compound is a 3 carbon compound. So with this we finish the splitting phase of glycolysis also. So till now we have finished 5 reactions involving glycolysis. So we have begun our glycolysis with phosphorylation reaction, and now we are in the I mean, till now we have seen two isomerization reactions, one phosphorylation reaction, and another mm, to for another a cleavage reaction. So now now we are entering into the final phase of glycolysis, which is energy generation phase. So you have to remember one important thing is, so energy is consumed in the first phase itself, hence we have named it as energy investment phase or priming stage. In that too, over the three reactions, only the first reaction, which is phosphorylation, and the other reaction, uh, which is also a phosphorylation reaction, we have observed the consumption of ATP. And so now the first reaction in the energy generation phase is, mm, oxidation reaction. So the glyceraldehyde 3 phosphate molecules, two molecules are formed. If you remember correctly, DHAP also reversibly converts to G3P in the presence of enzyme phosphostriose isomerase, right? So these two molecules of glyceraldehyde 3 phosphate undergoes an oxidation reaction. By oxidation here I mean, so there's no involvement of oxygen here. Here the oxidation reaction means a removal of hydrogen occurs. So in that way this is an oxidation reaction. So what happens is, for the first time, so NAD plus takes this hydrogen and forms NADH, um, and uh, one so from this G3P, so we observe formation of 1,3-bisphosphoglycerate is formed in the presence of enzyme G3P dehydrogenase. So this G3P dehydrogenase, so you have to so one important thing is, so this G3P dehydrogenase can be inhibited by iodate or arsenate. So what is the other important feature of this reaction is NADH is formed here. So in the presence of aerobic respiration, or if the aerobic respiration occurs, so this NADH is directly carried to electron transport chain, where each molecule of NADH helps or results in the formation of 6 molecules of ATP. So that's how important is the formation of NADH here. So the sixth reaction of overall glycolysis is an oxidation reaction, so which is mediated by, or which is catalyzed by, enzyme G3P dehydrogenase. So and the end product formed in this reaction is 1,3 bisphosphoglycerate. So now the next reaction, which is seventh reaction, is this 1,3 bisphosphoglycerate uh, is catalyzed to form, or is converted into 3-phosphoglycerate. That means we absorb a removal of one phosphate molecule here. So this removal of phosphate molecule is used for the production of ATP from ADP. In this reaction, ATP is generated by taking the phosphate molecule, which is removed from 1,3-bisphosphoglycerate molecule, and this reaction is catalyzed by phosphoglycerate kinase. So this is a reversible reaction. And see, you understand this. This is a rare example where a rare example of a reversible reaction catalyzed by a kinase enzyme. So the importance of this reaction is the ATP is generated from by taking the phosphate from the substrate level itself. That means the 1,3-bisphosphoglycerate is losing a phosphate molecule and donating it to ADP to form ATP. So hence this reaction is called substrate level phosphorylation, though the reaction generally named as dephosphorylation here, as the substrate as a phosphate molecule is being removed from the substrate. So this also is the best example of substrate level phosphorylation. So that means ATP is synthesized from ADP by taking the phosphate molecule which is liberated from a substrate. So this 3-phosphoglycerate enters into the next reaction, which is an isomerization reaction in the presence of enzyme phosphoglycerate mutase. So it reversibly converts to 2-phosphoglycerate. So 2-phosphoglycerate. In this reaction, what we observe is 2-phosphoglycerate. A magnesium molecule acts as a cofactor for the mutase enzyme here. Also, I guess there. So the next reaction is isomerization reaction is also done here. So till now we have observed 8 reactions in glycolysis. So the ninth reaction in glycolysis is a dehydration reaction. That means one molecule is removed from the substrate. So what is the substrate of this reaction? The substrate for this reaction is 2-phosphoglycerate. So 2-phosphoglycerate in the presence of enzyme enolase results in the formation of a powerful compound, a high energy compound called phosphoenol pyruvate, which is abbreviated as PEP. So this phosphoenol pyruvate is generated from 2-phosphoglycerate by removal of water molecule. So what is the importance of this enolase? Like a water molecule is removed and That's why the name of reaction is also a dehydration reaction. So now the next and final reaction is no, the another important point is this enolase enzyme is activated by two cofactors. One is magnesium ion or manganese ion. So the if you remember properly, so this conversion of 2-phosphoglycerate to phosphoenol pyruvate, so the enzyme is catalyzed by the enzyme enolase, which can be inhibited by addition of fluoride molecule. So fluoride inhibits the function of the catalysis function of enolase enzyme. So what happens generally is fluoride combines with magnesium cofactor here and phosphate and results in formation of a complex. So what does this complex do? This complex directly binds to the active side of the enzyme enolase, and it inhibits the space for or the active site for substrate molecule to act. So in this way, this reaction becomes a very good example for unusual competitor inhibitor. So who is the unusual competitive inhibitor here? So fluoride acts as unusual competitive inhibitor here. The other important point or thing we have to know from this reaction is, see, in general, fluoride is used in laboratories while testing blood glucose concentrations. So because, see, fluoride, when fluoride is added to blood, it inhibits the glycolysis reaction mm, in your blood. So that's why fluoride is a fluoride is what you call is a very good lab agent for us to measure or for us in getting proper results of glucose levels. So now let us move to the next reaction, which is the final reaction in glycolysis. The final reaction of glycolysis is mm, what do you call a phosphorylation reaction. We observe another molecule of ATP being synthesized here. So what is this reaction? This reaction is conversion of PEP, which is phosphoenol pyruvate, a high energy compound, being converted to pyruvate in the presence of enzyme pyruvate kinase. So what happened? So here another molecule of phosphate is removed from PEP and pyruvate is resulted. And the resulted product is of pyruvate. So here if you observe totally, so start the glycolysis has begun with a 6 carbon compound, which is glucose, and in the first three reactions itself we have seen a case where two molecules of ATP have been consumed, and later at the step of cleavage reaction, so 6 carbon, one molecule of 6 carbon compound is split and or cleaved to form two molecules of 3 carbon compound. Now, so you have to understand one thing is, starting from this phase the rest of the reactions or the five reactions in the energy generation phase occurs twice, because so each and every molecule of 3 carbon compound undergoes the same reactions. As we have two molecules of 3 carbon compounds, so all these set of reactions occur twice. That means the ATP generated overall in these from these reactions is a 4. So now if you observe the net result of ATP from a glycolysis is 2 ATP. So if anaerobic respiration occurs, glycolysis also results in formation of 2 into 6, 12 ATP if the NADH produced enters into the electron transport chain. From one molecule of 3 carbon compound we are getting one NADH, and if as the reaction occurs twice, or as all the reactions occur twice, we get two molecules of NADH, and these two molecules of NADH results in production of 2 into 6 ATPs, which are in the electron transport chain, which results in 12 ATPs. So the net we can call from glycolysis we get is net ATP we get from glycolysis if the process is an aerobic process is 14 ATP. So now we'll be looking at that. So with these 10 reactions I'll close the video for today. So let us look at into other aspects of glycolysis tomorrow. I mean where the glycolysis reactions can be observed in different pathways, how each and every molecule producing glycolysis can act as a initiator molecule or a branch point for other reactions. And let us move to the next parts of metabolism, various metabolism paths involved in carbohydrates. So thank you for watching my video. Please do subscribe to my channel Fuzzy Scientist. Till then, see you, bye, have a good day.